the mitochondria's
powerhouse itself.
But did you know this powerhouse actually doesn't synthesize most of its protein? Yes, most of the mitochondrial proteins are actually synthesized in the nucleus. So how did these end up in the mitochondria? Well, let's see how the mitochondria becomes the powerhouse of protein transport. I'm the Clumsy Shark. I'm currently doing my undergrad in microbiology and provide fun and educational chess and science videos. In the cytosol, the protein is translated and kept unfolded by chaperone. The protein knows to go to the mitochondria as there's a signal sequence that tells it to go to the mitochondria. This is the same thing as a mailman having address on a postcard. If there's no address, then the mailman doesn't know where to deliver the mail. The protein makes its way over to the mitochondria. At the outer mitochondrial membrane, there are receptors which will detect the mitochondrial signal sequence. Protein binds to the receptor, and the protein has multiple options. This is what the mitochondrial protein transport looks like. Yes, I'm not just going to slap it right out of your face, but let's break it down into the different components. So, let's take a look at our first protein translocator, the Tom complex. For any of you who are curious, this is what the actual complex looks like. So we can see that it's a lot of protein making up this complex, and we can see that there's a very little gap, so that's why the protein needs to be completely unfolded in order to be able to go through. The Tom complex translocates the protein from the cytosol to the intermembrane space for transport into the matrix, or it passes it along to the SAM complex. The SAM complex inserts beta barrel proteins into the outer mitochondrial membrane. One thing that's quite interesting is that it is actually similar to the BAM complex in bacteria, which reinforces the endosymbiote theory that mitochondria was an ancestral bacteria that got engulfed by a eukaryotic cell. MIM1 complex puts protein into the outer mitochondrial membrane. When the protein is being translocated across the Tom complex, the TIM23 complex can actually grab onto that protein and translocate the protein to the mitochondrial matrix. It will have its signal sequence cut off and will be fully folded with the help of chaperones, or it can directly insert it into the inner mitochondrial membrane. The TIM22 complex can insert proteins from the intermembrane space to the inner mitochondrial membrane. On the other hand, the OXA complex inserts proteins from the mitochondrial matrix into the inner mitochondrial membrane. It can actually insert proteins that are synthesized by the mitochondria itself too. Now you know how a mitochondrial protein that gets synthesized in a nucleus can end up in the mitochondria. Quick quiz time. Which of these complexes inserts beta barrel proteins into the outer mitochondrial membrane? If you were able to get it, comment down below
what the answer is.
Thank you for watching, and I'll see you in the next one. Peace out.